Okay, can someone confirm you can hear me?
I can hear you.
Great.
Yes.
All right, let's begin. Let me mention that the lab can be found in Canvas under week 1 lab. We're going to be covering lab module 00 today and worksheet lab module 00, as well as, no, that's it for today. Sorry, losing it. Any questions about any of that? If not, let me quickly talk a little bit about the syllabus, which hopefully you're seeing here. Can you guys see my page here?
Yeah, I can see it.
Okay. So the lab will be from 6:30 to 8:20. Generally, I only have the lab till about 8 o'clock. Excuse me here. I need a little bit of something to drink. I'm having a hard time in my throat. What happens with the lab is I'm going to talk generally before the lab about the lab, and then I'm going to open it up for you guys for doing the lab, and I will stay in Zoom to answer any questions. But by about 8 o'clock, I'll actually stop. Generally, students, when they log off, if all the students log off, I too will log off. And very seldom do I have a student staying till 8 o'clock. However, if a student does stay to 8 o'clock, I will stay here until 8 o'clock. Any questions about any of that? All right, let's talk a little bit about the syllabus, the parts concerning the lab. There is no textbook for the lab. There are some optional textbooks, but all of the material for you will be in Canvas, which I already showed you. You need for lab module 00, this lab module, as well as the worksheet. Any question about any of that? Now, the lab module itself has the lab, and then the worksheet is what you'll be turning in. If you turn in the lab module instead of the worksheet, it will have all of the text and will be much, much longer than the worksheet. The worksheet generally is a page or two. The lab module itself will be something like 7 to 12 pages. So if you turn in the lab module, even if you have all the questions there, you will get a penalty, because I don't want my Canvas website to become full and then students wouldn't be able to accept or turn in an assignment. So please only turn in the worksheet. Any questions? Generally, the questions are in the lab module, which is too bad that they are, but I've updated the worksheet, so it'll be the latest questions. All right, so only turn in the worksheet. How you turn in the worksheet is you will open this worksheet up. Come on. And you download it by clicking here or here. And then you can type in the document, the answers. And then you will have a link either on the right here or a link down here to upload it. I, of course, am an instructor, so I don't have the upload capability. I've got the download, meaning when you submit it, I'll be able to download them or hit the speed grader. You guys won't have these links. Any question about any of that? All right, so that's sort of how the lab works. Let me state that my computer is going to be kind of busy because we're still working on converting the lesson from the lecture. It looks like we're 93% done. But until that gets 100% done, this computer will be a little slow. It seems to be working at the moment. All right, you want to log in pretty soon at 6:30 because I've already started the lesson. Oh, wait a minute. Let me make sure I'm recording this. I do see a green button there, so I think I am recording. Once again, let me know in advance if you're sick so that we can get your work on getting an assignment turned in. If you let me know after you're sick, you will be turning in the assignment after it's due, in which case you'll get late credit. I got two of those open. That shouldn't be. Don't want to close that down because it'd probably close down the Zoom session. So I'm gonna have to deal with it until after the class, the lab. Get prepared for the lab. Generally, what you want to do is look over. If you don't read the lab before the lab, you should at least look over the lab. Now let's talk about late assignments. Let me mention that if you do turn in an assignment late, you'll have to email it to me in Canvas. I will accept it late, but you will get a late penalty. And all of this information for the lecture applies to the lab as well, but I'm not going to go over it again. I'm just going to go over things. We're not really going to use microscopes in this lab because everything we do will be done on the computer. So everything will be online. You will actually be using a virtual program where you will be using a microscope virtually online. So the microscope care specifically isn't so important, but you do need to know how to take care of a microscope, and you will be quizzed on that information. Any question about any of that? We'll go over how to use a microscope when we have a lab covering that material. All right, lab and lab safety rules. It's not so important because everything will be online, but you do need to know how to be safe in a microbiology lab. It's also a really good lesson for you, for all of you who are going into the health profession, especially nurses. Safety is one of the main skills they want to learn in this microbiology class. So safety is the primary importance of the lab, and never take shortcuts. Generally, only enrolled microbiology students and staff are permitted in a microbiology lab unless authorized by the instructor. All personal items should be in your book bag, your books, the purses, cell phones, coats. All of that stuff should be stored in a cabinet that we call a cubby hole. And that will be right as you walk in the door of the lab. Where the cubby hole is, as well as the door, we don't even consider that part of the lab. We consider that external to the lab. And I know that's a little confusing. If you have problems about that, I'll discuss it again. But my point is the door and the cubbyhole where you're storing stuff is not considered part of the lab. You then would walk into the lab, and everything that stays in a microbiology lab stays in the microbiology lab, meaning anything you bring into the lab has to stay in the lab, and it cannot ever leave the lab unless you were to get it sterilized. To sterilize it, you would have to put it in a disinfectant like bleach for something like 10 to 15 minutes. I think it's 15 minutes. Or you could have it autoclave, that's put it under a pressure cooker for 15 minutes. So if you did want to take like a penicillin and you wanted to take it home, it has to be sterilized before you can take it out of the lab. Any questions about any of that? So generally, everything a student has will be put in the cubbyhole, and that we don't consider part of the lab. And then when you leave, you can grab the stuff in your cubbyhole, and then you can leave the class. But if you bring it into the lab proper, it has to stay in the lab proper. Any questions about any of that? Paper and writing implements would be supplied by us, so you generally don't need things like that. Cell phones would not be used in the lab, and you just keep them in your backpack. Anything that is prohibited from being in the lab would be confiscated for safety reasons, and they'd only be returned to you after they've been sterilized. And for something like a cell phone, that would destroy the cell phone. You should realize that all cultures you'd be working with in the lab are potentially pathogenic. Keep hands, pencils, and other materials out of your mouth, ears, nose, etc. No eating or drinking or chewing or smoking or whatever in the lab. Furthermore, when you come in the lab, you put everything you want to put in your cubbyhole, like your books, your backpack, your phone, your purse, whatever. And then you go and wash your hands. Then you go and put on the lab coat. Then you go and put on the gloves. That is always required. Gloves would be supplied. The lab coat's generally supplied by us. A few students would buy a lab coat and then they'd bring it into the lab, and it stays there for the entire term. At the end of the term, it would then be marked that the students are going to want to pick it up, and it would be washed, actually autoclaved, then washed, and then the student could come and pick it up after the term. Closed-toed shoes must be worn in the lab at all times, meaning a lady's fancy shoes that are open, or sandals or thongs cannot be worn in the lab. The purpose for having a closed-toed shoe is to give your feet some protection in case of a laboratory spill involving a culture of bacteria. Tennis shoes do not exactly provide much protection, but they do provide some, more than something like sandals. Any question about any of that? And of course, the lab coat and the gloves are to give you protection too. And then at the end of the lab, you would, of course, you would first clean up your work area. We'll talk about that in a minute. And then you would take off your gloves, take off your lab coat, go wash your hands, and then leave the lab. So whenever you come into the lab, you put your things in the cubbyhole, you go wash your hands, put on the lab coat, put on the gloves, and then go to your workstation. And then when you leave the lab, you take off the gloves, take off the lab coat, go wash your hands, grab your things, and then leave the lab. Any question about any of that? Before you work in the lab, you would come to your lab bench area, and before you do any work, you sterilize the lab bench. And actually, you just disinfect it. It's not really sterilizing it. You use a disinfectant to wipe it down. You spray the disinfectant on, wipe it across the area with paper towels, wipe up the disinfectant, put it in the garbage can, and then you do your work in your lab space, lab bench. When you're all done, you disinfect the area you worked, and then you go get ready to leave, meaning you go take off your gloves, go take off your lab coat, wash your hands, and then grab your things, and then you leave. Any question about any of that? All right, there are some safety equipment in the lab, like you should know where fire extinguishers are, fire blanket, eye wash station, first aid material. You don't really need to know that because you won't be in a real microbiology lab, but you should know about the safety equipment. Any accidents should be reported to your instructor. Minor accidents should be cleaned as the following. Always clean broken glass first. Spilled strong acid should be diluted, then wiped up quickly. Spilled bacteria culture should be cleaned as the follow. Cover the spilled material with paper towels. Drench the paper towels with disinfectant. Let it sit for 15 minutes before wiping it up. Be especially careful with the broken glass. Use the brush or the broom and a dustpan to pick up the broken glass. Put the broken glass in the broken glass container, and then the paper towels, which have bacteria cultures on them, go in the biohazard trash can. Any question about that? We use bacterial incinerators. We used to use Bunsen burners. Keep your hair tied and away from the bacterial incinerators and the Bunsen burners. They should be turned off when they're not being used. Mouth pipetting is never allowed in the microbiology lab. All cultures should stay in the microbiology lab. They should never leave. You should consider all cultures as a biohazard and treat them accordingly, meaning if you get a spill on your hands, you don't suck your hands, which will have gloves or something like that. What you should do is wash your hands or disinfect your gloves because you got gloves on. Old specimens should be discarded in the following ways. Agar plates should be discarded in the biohazard container. Glass slides and cotton swabs have containers specifically for that, meaning we have a container for slides. It's usually up on the back of the lab, and you don't need to know that because you won't be in the lab. Cotton swabs, there's a place for putting them on your lab bench. Pipettes go in the pipette container. Glass test tubes go in the in a rack in the place for test tubes, and those would be autoclaved and then washed and reused. The slides too would be sterilized. You know, I don't know how, I guess they put them in bleach, and then they're washed and they will be reused. The cover slips are not reused and they would be discarded. They'd be sterilized and then discarded. All right, any questions about the lab and lab safety? Anything that has a bacterial culture on it that does not have any glass goes in the biohazard trash, meaning it's usually an autoclave bag, and we call it biohazard trash. You put anything that has bacterial cultures or contaminated by bacterial cultures in that container, unless it has glass. No glass ever goes in the biohazard container. If it's broken glass, it goes in the broken glass, what do you call that, container. Slides go in the slide container. Test tubes go in the test tube racks in the used section of for the test tubes. Any question about that? All right. So in the syllabus on the right, we have the lab schedule. We're going to be doing lab 0, lab safety training today. On Thursday, we'll be doing lab 01, ubiquity of microorganisms. Do realize you need to complete the online plagiarism quiz by 8 p.m. Saturday at Saturday, the 1st, January 7th. You must get 100% on this plagiarism quiz. And so if you want to retake it, make sure you take the retake or the several retakes before 8 p.m. For next week, you should be reading the infectious disease research project, because I'll be talking about that next week. And you can find that on Canvas. I'll talk about it on Thursday. Not talk about it, I'll show you where to get it on Thursday. All right, any questions about what we're doing today? If not, let's go to lab 001. I don't remember where I have that. I think these are the old ones. Yep. Give me a minute here. I'm not sure where I've got it. I guess we could always do it from the web. I've got that someplace on this computer, but I don't remember where I've got it. So that's what the trouble is. Well, I don't need to edit it. So lab module 00, laboratory safety. One problem with this is I don't have it highlighted. You're not going to be working in the lab, but you do need to know this material, as many of these rules will be relevant during your career. So the learning objectives are distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab, and that one's not so important for the online lab. Understand how to safely dispose of contaminated materials, and then where to put them. So microbiologists observe a number of safety rules as they're working with potentially pathogenic microorganisms. While you're not going to be coming into the lab and doing that, you do need to understand the safety precautions, including wear proper attire, such as shown here. You should have a clean lab coat on, covers your lap when sitting, it should be buttoned. You should have disposable gloves on. You should have closed-toed shoes. And if you're working with a splash hazard, you do need to wear goggles. The goggles are supplied by us. So are the gloves. And generally the lab coats. The closed-toed shoes have to be supplied by you. I think that's trying to show that the hair is tied back, the arrow there. Lab coats, goggles, and gloves must be removed before you leave the lab, and they would be reused. Well, not the gloves. The gloves would be disposed of. But the lab coat, you would use your own lab coat, and the goggles would be sterilized before you reuse them. So no food and drink in the lab, no use of electronics. No personal items are allowed in the lab, including cell phones. If they are brought into the lab, they would be confiscated. They would not be returned to you until they've been sterilized, and that would destroy anything that was electronic. So when you come into the lab, as I stated, you put your things away, you go wash your hands, and you do need to wash your hands properly everywhere for 20 seconds. I don't know if you can see, I'm trying to do that. Then dry your hands, go and put on the lab coat, go and put on the gloves. If you're going to be working with a splash hazard on that day, you would then go and put on the goggles. You only need goggles while you're working with the splash hazard. So you may wait until you're going to work with the splash hazard and then put on the goggles and then take them off when you're done working with the splash hazard. You come to your lab bench area and you disinfect it with a disinfectant and wipe it down. We don't have a spray that would necessarily go everywhere. We have a stream, so you have to spread it around with the paper towels. And this is actually a picture of our lab, what it would look somewhat like. Discard of waste materials in the appropriate receptacles. Regular trash, there's non-sharp objects, no glass, like paper towels and gloves that are not contaminated with bacteria, go in the regular trash can, which is not shown. It would be a trash can in the lab, and we have several of them. I think we have 4 in our lab. If they are contaminated with bacteria, you then put them in the biohazard area, and that would have a autoclave bag in it, and no glass ever goes in this bag, only paper and plastics that have been contaminated with bacteria go in here. That bag would be removed and a fresh bag put in place, and then that would be autoclaved before it would be put in the trash. Any questions about the biohazard and the biohazardous waste? Anything that has a sharp or glass does not go in the biohazard bag. We have a sharps container, which, to tell you the truth, we don't use sharps in microbiology. A sharp would be a needle or a scalpel, and generally microbiology students don't use those. If you did, you'd put it in a sharp container, and that's a little plastic box. You stick it in there. We don't usually use beakers, but if you do use a beaker, you'd put it next to the test tube. We do use test tubes, and you put those in the test tube racks for, I guess, the waste test tubes. Okay, and there's a spot for that, and that's where you'd put any beakers, which, like I said, students don't usually use beakers. Cover slips and slides go in the slide container. The swabs go in the swab, and these will be a container that have bleach or disinfectant in them. The slides, you don't need to know this, but they'd be washed and then reused. The cover slips will be taken out of the disinfectant, thrown away, and the swabs will be taken out of the disinfectant and then thrown away. Report all spills and accidents immediately. Cover with paper towel and then saturate the area with disinfectant and allow it to soak for 15 minutes. And then remove the area, the biohazard spill, wipe it up with the paper towels. No mouth pipetting, always use a pipetter. No chewing, like your fingernails or a pencil. No chewing gum or anything like that in the lab either. We don't use a flame, but we use a back to incinerator. That's a loop there, so that there. And what you do is you put the metal portion of the loop, the wire portion, inside the back to incinerator, and you see how that's red hot, and then you sterilize it. You then let it cool before you picked up the bacteria on the loop. And then you put the bacteria where you want it to be. So you only use the bacteria incinerator, have it on when you're using it, or right before you use it, because it does take 5 minutes to warm up. And then when you're all done using it, you turn it off. So these are some safety stuff in the lab. A fire hydrant in case there's ever a fire, an eye wash station in case you get something in your eye. I think that's a fire alarm. Hopefully we won't ever get that. A fire hose, probably a fire blanket up there. Oh, there's the fire alarm. So what's that? Don't know. A deluge shower, you pull this down and then that spreads water all over the floor. But if you were to get like acid spilled all over you, you want to step under the shower. Never had to use that. In fact, I've never had a student have to use that. Although many years ago, probably over 20 years ago, I did have not a student, a faculty member have to use the eye wash station. Any questions about the safety equipment? All right, so read the lab modules before you come to class. When in doubt, always ask. Please complete the laboratory exercises in the worksheet, a separate file. Lab procedures and safety rules, never take shortcuts. You're given a procedure, please follow that procedure. I already talked about that. Already talked about that. Talked about that. The gloves are non-latex. They must be worn while you're working in the lab or when you're in the lab. If you wear something like thongs or sandals or women's open-toed shoes, and you come into a microbiology lab, you would have to change your shoes, which is allowed, and then put your closed-toed shoes or sandals in the cubbyhole. Goggles do need to be worn when you're working with a splash hazard. A splash hazard would be something like a liquid culture, which could splash bacteria into your eyes. All right, I've already talked about all of these other things. So, here's one I haven't talked about. If you have a cut or a sore on your hand, you should come into the lab, put your things in the cubbyhole, wash your hands normally, and then go over to the first aid kit, grab a Band-Aid and put the Band-Aid on. And then put the glove on if it's in your hand, put the glove on over the Band-Aid. All right. Any questions about that procedure? All microbiology cultures you work in the lab must be completely labeled with your name, the date, the nature of the specimen. That would be something like, if you know what it is, state what it is, E. coli, for example. And then the medium used. It is optional to put the temperature that you grew the microorganism on. I already talked about disposing of material. Supplies needed from the lab would be supplied in the lab. All right, any questions about any of that? If not, go ahead and begin working on the lab. Let me try and get there. Open up the worksheet, lab 00. Do I have that open? Yeah. Download it. Yeah, there it is. All right, the worksheet does not display properly, at least in the computer. And surprisingly, when people print it out, there were some questions that showed up. I don't know what's going on with the worksheet. So you can ignore the first page and just skip to the second page where I type the questions. So answer these questions. Rule 1, in the lab, what do you always wear for safety before you enter the lab? It has to be before you enter the lab. And there's 4 asterisks, so you need 4 things. Okay? Rule 2, after you enter the lab, what do you put on to keep yourself safe in the lab? And this is something that you do not have on before you get into the lab. And it's not until you enter the lab, usually after you wash your hands, that you would put this on in the lab. Okay, so what's in rule 1 should not be in rule 2, and what's in rule 2 should not be in rule 1. Do you need any other hints? I'm not hearing anything. Is there somebody there?
Yes.
Yeah, I'm
Great.
here.
All right. All right. For rule 3, you have to look up this information online because the lab manual does not discuss it. Look up what is a safety data sheet online. And then for each chemical in the lab, you must have a safety data sheet, an SDS, that describes what? And you have to label 1, 2, 3, 4, 5 different things. Generally, you cannot have a single word. You have to describe what is on the safety data sheet. Okay? Any question about that? Doesn't have to be a sentence, but you do need at least a phrase. 4, know the location of your safety equipment, such as, and then you just need to label something that's a safety equipment. What is a safety equipment that was mentioned in the lab module? You need 5 different safety equipment. What was the safety equipment? Come on. Somebody knows.
A lab jacket.
You're putting that
First aid
on for
kit?
safety, but we don't consider that safety equipment.
Would a first aid kit qualify?
First aid kit would qualify. A fire
Fire extinguisher.
Yeah, fire extinguisher is what we're talking about.
The
But
shower.
I only asked for one, so thank you. Okay, don't give everyone all the answers. But a lab coat is close, but that's something that is required of all students at all times in the lab. And the safety equipment you would not need unless an emergency happened, like you'd have to put out a fire or something like that. Okay. So that's what we mean by safety equipment. What can you take into the lab? So that would be like the first one. And then can you eat and drink in the lab?
No, you can't.
No.
All right, that's fine. Yeah, cannot eat or drink in the lab. And then list one item of laboratory attire that you must wear in the microbiology lab. And this one can be from either the same as rule 1 or rule 2. And then list one item that is not allowed in the lab.
Phone,
Sorry?
phone,
Yeah, you can't take the phone in the lab proper. And then I might have missed question number 3. List one item that is allowed in the microbiology lab. And this would be something you bring from outside. Students always have trouble with this. So I'm going to say you can bring in jewelry, you can bring in your watch, you can bring in a necklace, for example. So that's what we're getting at with 3. Submit this file by 8 a.m. Friday of week 1. So worksheet lab 00 must be turned in by Friday at 8 a.m. or you will be dropped from the class, meaning I'm taking this worksheet for attendance to determine who is in the class, who enrolled for the class. And if someone does not turn in the worksheet by Friday of 8 a.m., you will be dropped from the class. I have to turn in the drop list by Friday. I think it's 5 p.m., but they may have upped the date, the time. It might be 1 p.m. I don't know. That's why I have it that it has to be turned in by 8 a.m. Any questions about any of that?
No.
If there's no questions, go ahead and get started on the lab. I will be here until 8 o'clock to answer any questions or until the last student logs out from the class. When the last student logs out, I too will log off. All right, get started on the lab.
I have a question.
Uh-huh.
For the submission, it says that we could do text entry, and I wanted to make sure with you that that is, in fact, an option.
Let me check.
Because how I did it is I answered the questions in text entry format instead of on the actual document, if that makes sense.
I understand. Let me check to make sure. Oh, it's right here. Edit. Let me see what it accepts because I don't remember. Yeah, I'm allowing a text entry and a file upload. Your text entry will be TXT. Now, when you put in the answers, make sure you have questions. So I know
Okay.
what you're answering.
Okay, so it's okay for me to type in my answers as long as I include the question in there?
Yeah, like I said, I'm allowing, I don't know if you can see this. Let me display this. There, I'm allowing a text entry as well as a file upload. So you can type in the text directly in, I don't know, the Canvas webpage and then upload the text.
Okay, awesome. Thank you.
Okay, all right.
So the remainder of this lab is only for you to answer questions on the packet, right? For the Zoom. Right now, are you just here to answer questions if we need help?
Yes, from now on, I'm here just to answer questions. I'm not going to talk any further. Oh, why I
Thank
should
you.
is I should stop recording this.